Based on a study published in Nature Aging and led by Professor Francesco Neri, scientists have identified a systematic age-related epigenetic process in the gut that significantly increases the risk of colon cancer. This process, termed ACCA, Aging and Colon Cancer Associated Drift, reveals a hidden biological program behind intestinal aging. The lining of the human intestine is constantly renewed by specialized stem cells. Over time, these stem cells accumulate epigenetic changes, chemical tags on DNA that act like switches to turn genes on or off. The key discovery is that these changes are not random, but follow a predictable pattern that intensifies with age. The genes most affected are crucial for maintaining tissue balance, many of which are involved in the WNT signaling pathway essential for intestinal renewal. As these genes are progressively switched off through excessive DNA methylation, the gut's natural repair mechanisms weaken. Critically, this same drifting epigenetic signature found in aged gut tissue was also present in nearly all analyzed colon cancer samples. This strong overlap suggests that the accumulated changes in aging stem cells create a cellular environment ripe for cancer development. Aging does not affect the gut uniformly. The intestinal lining is composed of millions of tiny units called crypts, each originating from a single stem cell. When a stem cell acquires epigenetic changes, every cell in that crypt inherits them. As explained by Dr. Anna Krepalova, through the natural division and expansion of these crypts over years, patches of tissue with an older epigenetic profile grow and spread. Consequently, an older person's intestine becomes a mosaic of relatively healthy crypts that are epigenetically aged and prone to generating damaged cells. The research team pinpointed a specific molecular cause for this drift: iron dysregulation. As intestinal cells age, they take up less iron and release more, leading to a depletion of iron, Ti, within the cell nucleus. Iron, Ti, is a vital cofactor for TET enzymes, which normally function to remove excess DNA methylation marks. When iron levels drop, TET enzyme activity falters, faulty methylation marks persist, and key genes are permanently silenced. This failure of DNA demethylation accelerates the epigenetic drift. This process is further exacerbated by age-related gut inflammation. Even mild inflammation disrupts cellular iron balance and metabolism, while simultaneously weakening WNT signaling. The combination of iron imbalance, inflammation, and reduced WNT signaling acts as an accelerator, meaning intestinal aging may start earlier and progress faster than previously thought. Despite the complexity, the findings offer promising therapeutic insights. In experiments using intestinal organoids, miniature gut models grown from stem cells, researchers could slow or partially reverse epigenetic drift. They achieved this by either restoring cellular iron uptake or directly boosting the WNT signaling pathway. Both interventions reactivated the TET enzymes, allowing cells to clear the problematic DNA methylations and reactivate silenced genes. As Dr. Krepalova notes, this demonstrates that epigenetic aging does not have to be a fixed final state. For the first time, it appears possible to intervene in the deep molecular parameters of aging within cells, potentially opening avenues for strategies to delay gut aging and reduce associated cancer risk by targeting iron metabolism or epigenetic maintenance pathways.